This is case 16, a piercing gone wrong.
Our patient's name is Sierra, a 17-year-old female. She was admitted to the hospital for symptoms such as fevers with chills, nausea, vomiting, and diarrhea. The cause was due to an unprofessional at-home nipple piercing. The patient didn't tell the doctor, so they proceeded to do an examinations and tests. Upon the physical examination, the doctors viewed her body and noticed that the torso had a rash and slight discharge coming from her nipple piercing site. Yet, since they didn't know that it was due to that, they proceeded to do more tests. Oh my God, a sec. And they moved on to a pelvic examination and found that that was the root of the problem. These are the laboratory results. And here it shows that either some were high or some were low, but in all cases, due to the results, all of them were high. These were also shown to be high. And this just proved that her body was fighting off an infection. So after finding out the evidence to show that she was fighting off an infection, they put her on a medical ventilation, they gave her IV fluids, and then they also treated her with 3 different types of medicine. And then they were mainly treating the skin rashes and infections, as well as the pelvic inflammatory disease. The reason for treating the pelvic inflammatory disease was because the pelvic ultrasound showed that she had a complex left mass and there was pelvic hemorrhage fluid. And then after changing the treatment to treat the pelvic issue, her condition worsened, and upon being admitted to the hospital, she died within 36 hours. So after her death, the doctors looked at her and found that her breast was the problem. And then here's a picture of the ultrasound just to show what it would have looked like.
So here is also some of her results. This is basically just looking at the VB2, and we'll be talking about that later, but it's involving with the superantigens and the MHC cells, which we'll talk about in the next slide. But superantigens bind to MHC cells, and then those bind to variable beta or alpha chain of T cell receptors, and it can bind to multiple variable beta regions, the VB, like here we see VB2, VB8, VB5, VB12, and same kind of up here. And so we can see in both of these, she shows a really high VB2. And so in these figures, in this one, this left region here is a kind of like a control sample. And this is just kind of where it is in the immune system. So you can see how many of the cells are there. And here you can see this is pretty low over here, but then this is Sierra's over here, and around this area, it shoots up really high to close to 80 cells of the VB2. And then also down here, and so you could see how this is obviously abnormal. This is a high amount of VB2 over here. And then on this one, on this left figure, we can see that the VB2 in Sierra's sample right here is also really high because it's supposed to be kind of like this like small dot, and it's this very large dot over here on VB2. Her VB8, VB5, and VB12, they aren't like showing up much, but it's just this VB2 that we're like kind of focused on. And so our first question is, superantigens primarily activate CD4 T cells, which MHC cell molecules would you expect to find superantigens bound to, and why is the binding to the MHC faster than that of most antigens? So going back to what I was talking about with those VB2 samples of Sierra's, to explain it all, superantigens are antigens that result in excessive activation of the immune system, so kind of like an overproduction. And they are bacterial toxins that generate a lot of an immune response. So as we see in the diagram, The super antigen binds to the antigen presenting cells, which is this APC yellow region right here, and to the T cell receptors, which is this blue region right here. And they induce an excessive amount of cytokines. And also, this is probably why she had all this rashes from the excessive amount of cytokines. And then MHC cell receptors are abbreviated for a major histocompatibility complex, and they are the cell surface proteins that help recognize foreign substances in the immune system. And there's two types. There's class 1 and class 2. Class 2 deals with CD4 helper T cells, which is stated up here. That's primarily what they activate. And then there's also type 1, which reacts with CD8 T cells. And normally the MHC would have a groove that the antigen binding groove, and then it would bind to the protein and then to the T cell receptor. So in this, the first question, what would you expect the super antigens to bind to? We would expect it to be class 2, which is the CD4 helper T cells. And then why is the binding of super antigens to MHC faster than that of most antigens? The super antigens holds and activates receptors in MHC regardless. So like whether the shape or size or whatever. So it's going to have an overproduction of the T cells that are being activated, which is those cytokines, that being an abundance. So the high number is activated because it's not specific to what it's binding to. So it's just binding to a bunch. And that's why it's going to be faster.
Question 2 asks, how would the response to a super antigen be different if it stabilized interactions with MHC class 1? Class 2 helper T cells are present with MHC CD4 and they interact. When they interact, they create interleukins. When class 1 cells are present, MHC CD8 interacts and creates cytotoxic effects. Therefore, stabilizing with CD8 instead of CD4 will lead to increased cell death in the area.
So our next question says, activating CD4 T cells normally leads to activation of B cells, dendritic cells, and monocytes. Why didn't this happen in Sierra? So basically, what the cells have in common is that they are all antigen presenting cells, which we have shown here on the slide. These aren't being made because they are being mobbed by T cells. So normally there will be one MHC with one T cell, and the super antigen, it is interacting with tons of other cells, so it is stuck and can't do anything more than, and like anything, nothing else is being made, basically. So it's basically saying like if you have one supermodel and 50 guys versus one supermodel and one guy, the supermodel with 50 guys can't really do anything.
And so the last question, TSST1 is what Sierra ultimately had, which is toxic shock syndrome toxin 1. And basically what that is a super antigen that causes toxic shock. And this is what released the high amounts of her interleukins. And then this is also a part of the super antigens that creates a ton of cytokine production, which we talked about earlier. So this is an exotoxin, and it's stated right here in the question. It's secreted from bacterial cells while the bacteria are still alive. So how would you expect the illness to be different if the bacteria produce an endotoxin? So with exotoxin, we already stated the bacteria can only produce while it's alive. So you would treat it with an antibiotic that would kill the cells. But with an endotoxin, the bacteria is not produced when it's alive. So if you treat the patient with an antibiotic that kills the bacteria, you'd kill it too fast, you'd end up producing more of it. So you want to put them on something that will limit the division of the bacteria, but not just kill the bacteria. So basically you would just be treated with a different type of antibiotic.
So the ER doctors were flawed in their approach. They did a pelvic exam and were focused on abdominal exams. They should have focused on the rash associated with the nipple piercing. Normally, toxic shock syndrome toxin comes from the pelvic area. The ER doctors focused on that and ignored the fact that the problem came from the piercing. It is known that tampons cause toxic shock, so the focus was on that. In the end, what Sierra had was toxic shock syndrome toxin caused by an infected piercing. This was a major error the ER doctors made, focusing only on the pelvis. That evidently resulted in the death of 17-year-old Sierra. The autopsy showed no evidence of infection in the pelvis. It did show right breast inflammation and a small amount of pus deep to the piercing site. Hopefully, knowledge was gained from this, and this mistake will not be made again in the future.